Okay, so if I were trying to study for the biology test, I would use my eval sheet to kind of help. And I can send this to you, okay, to go through. So this is what I would do, okay? So what I would do is I would start going through my papers. So one of the things is while we are doing, hold on, there we go, a little bit better. What I would do is while we are doing our unit, I would make sure that as we're putting stuff in the table of contents, that's the way it's going in my folder. Okay, so for example, and I know you had this in junior high school as well. So take this lab first, the cell surface lab. And can you talk about this lab and the parts? And maybe we need to make note cards off from some of our activities. So for one thing, cell size comparison and diffusion. That was with the agar cubes. So I'm like, okay, agar cubes. So with the agar cubes, we looked at the bigger the cell is, okay? And this is not on the test because this was on the last test. So you really don't have to worry about it, but think about, we did this lab because we were learning those different concepts. So you're like, we did those because, the cell got too big and then we needed to divide. So that showed the bigger we get, that ratio. Okay, that's not on this test. Remember, that was before exams. But that led us into, if we're too big, then we divide. So then we start talking about why we're multicellular. That is also not on the test. That was on the previous test. But that led us to mitosis, division, cell division. So I know that we made some note cards on this, right? So this is our set of notes, and I will always give you guided notes. So totally make note cards off from this, okay? This is cell division. And remember, we went on the back side for the test because we talked about DNA replication, but that was before the exam, okay? So then we went on and we talked about all the different phases of the cell cycle. Then after we did notes, we did a lab. Remember, we were looking for phases of mitosis. So that was what this was all about. But we also did an online piece. Remember this? This was that Arizona piece where we had to count how many were in each cycle. And the whole part about this was, again, let's go and apply the lab. So the other lab was about the phases. This lab was about the cell cycle. And so this told us that the cell spends most of its time in interphase because there's so much stuff going on during interphase. So in my brain, I want to be thinking, interphase. Oh yeah, that's the G1, that's the S, that's the G2 cycle. And then after we did that, remember, we worked on some CERs. In fact, I told you the one CER was anaphase was the longest, when you all proved me wrong, right? According to our data, anaphase isn't the longest, it's interphase, okay? So then we did a movie and we filled this out together, okay? And this was all about the checkpoints, okay? So this was the G1, and then we stopped. We said, okay, are you going to divide? You're going to differentiate, or you're going to die, okay? That was the first one. Then we go through DNA synthesis. We're going to replicate. We're going to divide. Okay, we're going to divide. Then we're going to multiply. We're going to duplicate our organelles. And then we're going to stop. And we're going to check. Did you replicate correctly? If you didn't, you're done. So remember, all those regulatory things there, okay, are gene regulations. And that's what started us talking about gene regulation. Like those are all things that are regulate. And then once we get into, we got prophase, okay, and then metaphase, anaphase. And right there at the metaphase to anaphase is another stop. Hey, did they separate right? Did those sister chromatids separate right? So separate and then go into telophase and then cytoplasm, cytokinesis. Okay? So that was what that worksheet was all about. And the back was all about these little proteins called cyclin and CDK. So remember that CDK is the type of, it's kind of like the dominoes, like you have to set up dominoes, but somebody's got to flip over the first domino, right? That's what cyclin is. So the cell won't go into splitting until these cyclin levels, these proteins come up, and then the cell knows. So we're talking about regulating the cell cycle. When does a cell divide? Well, it's regulated by growth factors. Is there enough elbow room? If there's not enough elbow room, like, hey, you're touching me, this cell, then the cell won't let division happen because there's not enough room, okay? Or if the cell is damaged, then it won't allow it to go on. So it's kind of like a stop point that, hey, maybe you don't need to go on, okay? Because there's not enough room for you to divide. So that is what that is, a cell cycle regulator. So this sheet and this sheet go hand in hand. And then remember, what can go wrong? Cancer. So cancer is what we talked about down here, is that there's nothing stopping, okay? That something has gone wrong with the regulation of the cell cycle. So nothing's stopping it. It just keeps growing and growing, which is how we get tumors, okay? And lots of things can do that. So lots of things can make our cells go wrong. Like in the book, it talked about, remember we read that it talked about tobacco, smoke, or other chemicals that we're exposed to, or alcohol, or mutations, or viruses like HPV. And that's what got us started. Remember, we talked about cancer, and we watched that great movie on Henrietta Lacks. Remember? And she had cancerous stem cells that they took from her body without her permission. And they just kept growing and growing and growing. In fact, they're still alive today, which is amazing, right? That we are actually able. So that's where, remember, we are watching that video. Maybe that's a video you want to show or rewatch. You can always go to Plan Book. So we looked at Henrietta and why cancer and how it was very prolific. Unfortunately, we know she died of cervical cancer and of the treatment that she received. And she was treated only in a certain portion of the Johns Hopkins Hospital because there was still segregation at the time. Just another practice. Remember the mouse on independent, variable, dependent, writing a reasoning, a claim. And then we talked about those cell regulators. All right, well then how do we differentiate? Because a cancerous stem cell can be anything you want. So the DNA, remember, differentiate is that DNA says, okay, you're going to be a bone cell, and you're going to be a hair cell, and you're going to be a nerve cell. That's what we were, in fact, this cell right here, this was an intestinal cell. So this intestinal cell's job is to produce, for example, lactase, okay, which is why we were talking about that. But everybody's DNA was the same. It's just what DNA was turned on. That's exactly what we needed to get to, right? Because our girl could not digest milk, okay? Our girl couldn't digest milk. How come? Well, we all, as adults, can't tolerate milk. The majority of the population, nobody should be drinking milk as an adult because we're the only goofy mammals that do that, okay? So most adult mammals don't drink milk. They only drink milk when they're young because that's the only food source that's available to them. But in the human population, there is a mutation on chromosome 2, okay? And we watched that video of Got Lactase. There's a chromosome mutation that occurs that allows people, there's a switch that keeps producing lactase. It's almost like a machine that just keeps going for some people, okay? I'm not one of them. There's that machine. So in our culture, so if you want to rewatch that video, in the culture at the time when we were hunter-gatherers, nobody really drank milk, not a big deal. But then during a time, they became herders. And when the crops died off and they had to rely on their milk from their cows, if you couldn't produce lactase, then you were getting diarrhea and a stomach ache, okay? And then you weren't passing on your genes. So that's how culture and biology worked hand in hand, because the mutation was already there. Culture just pushed those people that had that mutation to be more successful at the time. So in class today, we did a little simulation where I marked everybody's hand with pink, or I left you, and we partnered up, and then we went through, like, say, hey, we're doing really well, and everybody rolled the dice, and we all had children, and some were pink, and some had the pink, and some didn't. And then I said, we had a famine, so we have to rely on milk. So everybody who doesn't have the pink, you just died. And so the population changed. And that's exactly what happened in history and led us to lactase persistent. But right now in our society, we don't select against those that are lactose intolerant. So even though certain students in our hour are lactose intolerant, there's other choices for them, and they're not getting selected against. So it's not really a big deal in our culture right now. So that's why you're not seeing maybe more lactose persistent people, because there's no reason. It's not making them more successful than others. Okay, I kind of went through the rest of the packet, but that's exactly kind of what I was getting through. Okay, so again, here's our notes on how the lactase breaks up. Okay, that breaks up the lactose. But then the next few days, we spent going through our lab. How does your notes apply to your lab? Well, the lab was all about lactase and breaking up lactose. Okay, and we tried different types of milk, and then we tried it in different temperatures and pH to show you that enzymes are very fussy. They have to have optimum conditions. So in the stomach, in the gut, it wants it to be warm, and it wants it to be acidic because it's from the stomach. And so those are optimum temperature and pH conditions for an enzyme, because an enzyme is very three-dimensional. And if it starts to change its shape, and then the enzyme can't fit, or the enzyme can't fit with the substrate, then it's not going to get broke up. So sometimes you can get too hot, and then it's kind of like the dishwasher analogy, remember? Okay, so that's not really going to work. So the rest of the papers in here, again, go back to this lactose. I'm not interested in the specific stuff. This is just helping you understand the basic concepts of how does lactase work. Do you know your vocab? Do you know substrate and enzyme? And do you understand what the process, what hydrolysis means? That's the piece. And so the rest of this is just helping you kind of reinforce, again, going back over this worksheet with the enzyme. It's just literally going back over your initial notes, okay? Going back over your initial notes, okay? So it's all related together, I promise you. It's all related together. I'm not looking for specific details. I never have, okay? But I'm looking for those concepts, and I'm doing activities to reinforce those concepts and try to get your head wrapped around those concepts. So maybe when we're doing those things in the laboratory, that you're remembering those concepts from that notes to make it a little more real. There won't be any metric system on this test, but I did ask you a couple percent questions because we had trouble with percents, remember earlier with the Arizona. So just multiplying percents, but you can use a calculator. So hope that helps.